everyone. Okay, it's recording. Awesome. Today I will talk about this study that I developed during my postdoc at the Highland Lab under supervision of Dr. Gregor Goriank. And it's about temporal and genomic analysis of additive genetic variants in breeding program. This study has recently been published in the Heritage Journal. So if you want to have a better look on that, you can check out here. Okay. First of all, I want to start this presentation just summarize the main points of this study, this publication. Here we develop a three framework to analyze genetic variance and its component for different stages of a breeding program, which we call breeding analysis, over time, temporal analysis, and by chromosome and loci level, which we call here genomic analysis. Well, I stole this slide from Gregor because before presenting the paper, I want to show you why we should be interested in analyzing genetic variance. Well, at the Roslin Institute, the Highlander Lab group collaborate with a number of leading breeding programs, and they want to develop a set of tools that would enable them to assess how much variation is there in a breeding program at each time point and in a different genome regions. So basically, they want to ensure that we prevent situations where a well-functioning breeding program is outcompeted by other breeding programs that is managing its germplasm better, as we can see here for the blue versus the pink breeding program. So how we analyze genetic variance for different group of individuals or over time if we assume that we know the breeding values? We would simply partition in this vector of breeding values and calculate variance of these values. This partition can be in year level or can be in a stage of the breeding level. So here this will give us the breeding and the temporal analysis of the genetic variance. If we go further and assuming that we know the causal loci, we can also partition each variance by the causal loci. This genomic partitioning here would give us the information of genic variance, that is a function of genotype variation at an individual loci and the allele effects. For example, here the variance of this green with itself. Then there is also covariance between loci on the same chromosome. For example, here the loci green with the loci pink in the same chromosome, which is due to linkage, and finally covariance between loci on different chromosomes. For example, here green and orange loci, which is due to linkage-disequilibrium. So the sum of these three components, genic variance, within chromosome linkage disequilibrium, and between chromosome linkage disequilibrium, give us the genetic variance at a particular time point. Well, in reality, we never know the true breeding value, so we need to estimate them from the data. Traditionally, we use these pedigree-based models, where we can estimate base population genetic variance by fitting a model to the data. This variance actually is a parameter in the model. In 2001, Sorensen and collaborators, they present a method for temporal analysis of genetic variance, which consists of fitting a pedigree model, sample breeding values from the fitted model, and then summarize these sample values by some time partition to get genetic variance by time. These steps you will see, we will repeat a lot in this presentation because it's the main steps to partition genetic variance in different levels. Currently in the genomic era, we have many proposed models and methods to estimate the genomic and genetic variance. As an extension of Sorensen's studies, we effectively, again, the steps, fit a market-based model, sample mark effects from the fitted model, then from the sampled mark effects and genotypes, we calculate the samples of breeding values, which we can summarize by time partitions. And if we want genomic analysis, we can also calculate breeding values for genome partition and calculate variance for these partitions. So in summary here, the main goal of this study was to demonstrate a framework for temporal and genomic analysis of genetic variance in breeding program. As said before, this framework fit a market-based model to the data, sample realizations of mark effects from the fitted model, and for each sample calculate realizations of genetic values, and finally calculate the variance of sample genetic values by breeding stage, time, and genome partition. So our study added to the literature by demonstrating temporal changes in linkage disequilibrium under selection, both within one breeding cycle and between breeding cycle over the years. Regarding the simulations, we simulated a realistic wheat breeding program with wheat genome and selection on yield for 21 years. We simulated this wheat breeding program using the AlphaSimR program. And we also followed the breeding program described by Gaynor and collaborators. In the breeding program, we created 17 inbred lines, and we random crossed them to generate 100 F1 that had the genome doubled, give us 10,000 head rows. At this stage, we simulated visual selection, here with a low trait heritability of 0.1, and we advanced 500 best plants to these preliminary wide trials. These trials means that the plants were evaluated in an unreplicated experiment, just one location, one repetition, considering a trait heritability of 0.2. As we advance the trials, we also increase the number of repetitions from 1 to 16, and therefore we also increase the trait heritability. So in the advanced wide trial, we evaluated the lines in a small multi-location replicated trial with 4 repetitions, and the trait heritability was 0.5. In the elite-wide trial, we evaluated the lines for 2 consecutive years in a large multi-location replicated trial with 16 locations, 16 repetitions, and the trait heritability was the highest one, was 0.67. By the end of the breeding program, just one variety is released. We used these best lines for advanced wide trial and elite wide trial as parent to create a new breeding cycle. And we run all these stages of the breeding cycle in parallel, so in this way we are allowing them to occur simultaneously in each year. Here, for example, in year 19, we have 7 breeding cycles occurring at the same time, with each one of them in a different stage of the breeding program. So what we are calling here breeding cycle is basically this diagonal line here, where we have the parent in 1 year, which generated the F1 in the next year, and go on. For our training population, we collected individuals from the last 3 stages in these preliminary, advanced, and elite-wide trials for the last 6 years. So it's in this gray square box here. Here we assumed that these individuals, we have data available for them from year 16 to year 21, which give us 3,420 phenotyped individuals that were also genotyped with 10,500 markers. Well, this is the statistical model we fitted to data. We assumed that marker effects are a priori uncorrelated and normally distributed. We also assumed uncorrelated and normal distribution for residuals. So with that, we are ignoring that different wide trials had different amount of replications and therefore should have different error variance. Here, a future study extension would be to account for this different error variance in the statistical model. We fitted the model using the alpha-based software, but you can also use other softwares like GWAS, BaseR, or BGLR to fit this model. We saved 1,000 MCMC samples of marker effects, and for each sample, we calculated the breeding values from which we then calculated the genetic variance for different breeding groups and different times. So here, in this phase, we partitioning our genetic in different stages according to the data that are taking account in this W matrix here. For genomic analysis, for partitioning of genomic analysis, we can see here, for example, a big picture of the genome. So this is a variance and covariance matrix. And we have here the chromosomes, and each dot here could be, for example, KTL in simulation analysis, but in real life, we do not know the KTLs in the genome, so it also could represent the SNP markers, for example. So each dot here is a loci in the genome. And we have the whole genome genetic variance in green. We can partition this genetic variance into chromosome genetic variance plus the between-chromosome linkage disequilibrium here. So it's in yellow and in orange. Then we can go deeper and partition in this yellow part into genic variance in the diagonal and within chromosome linkage disequilibrium of diagonal. So basically, genic variance refers to locus variance. Within chromosome linkage disequilibrium refer to covariance between loci in the same chromosome, and between chromosome linkage disequilibrium refer to covariance between loci on different chromosomes in this part here. So adding some equations here in this genome partitioning. Again, we have the whole genetic variance, and we can partition that in 2 levels. The first level, again, it's just split that into chromosome variance in yellow, and the sum of covariance between chromosomes in orange. Then we can partition in this chromosome variance in the second level in genic variance and in within chromosome linkage disequilibrium. So we end up with this equation where we have that genetic variance is equal genic variance plus within linkage disequilibrium plus between linkage disequilibrium. For the results, here I'm showing posterior distribution for genetic and genic variance for different groups of individuals with this variance in the x-axis. This is the genetic variance in one breeding cycle. So basically, is this this value here from parents from year 16 to elite white trio in year 21. So this is what we are seeing in this graph. The small vertical line denotes the true value, and we can see that the posterior distribution covers this true value nicely. We can also see that the parents at the top had the highest variance. Then, as expected, the variance dropped because parents in wheat are inbred, and crossing them create F1 individuals that collectively manifest less variation. This variation indicates variance in parent average between crosses. However, when we generate double haploids, genetic variation was stored to the level of the parents due to recombination and complete inbreeding. In the next stage, we reduced the number of genotypes due to selection, and during this process, the genetic variance is expected to decrease as well. We observed, we have not observed actually major changes in genic variance, suggesting that allele frequencies have not changed much. Of course, these genic variants are calculated for all loci, and extension research is needed to study changes in locus-specific genic variance. Now here I'm showing the temporal analysis. So I'm showing the evolution of genetic variance over time for different groups, and you can see that the posterior distribution with a 95% interval again match the true values in the solid lines very well. So the method here seems to be working well for temporal analysis using genomic data. We can also see that the most change in genetic variance over time is largely driven by changes in linkage disequilibrium and not in genic variance, because genic variance was quite stable over time. Furthermore, the variability of genetic variance over time increased in the late breeding stages as the number of individuals decreased. So remember, we started in the parents with 70 inbred lines, and by the end we finalized with just 10 elite lines here in the elite white trial. So less individuals, more variation in more genetic variation in the genetic variance. About genomic analysis, here we performed this genomic analysis partitioning again the genetic variance into genic, within-chromosome linkage disequilibrium, and between-chromosome linkage disequilibrium. And again, the posterior distribution matched the true value in this black line nicely. We can see that the variance dropped between the early stage of hetero in dark colors to the late stage of elite white trials. Remember, between these stages, between these heteros and elite white trials, we was performing selection. The genomic partition shows that this drop was in part due to a slightly decreased genic variance, either because we lost some alleles or more likely allele frequency just shifted. But more importantly, we can see that the substantial change in the between-chromosome linkage disequilibrium was the main driver in change of genetic variance between hetero and elite white trials. So this negative LD is a function of genotype combinations between loci, as well as the allele substitution effect. And it corroborates with Boomer when we affirm that linkage disequilibrium is an important component of the genetic variance. Well, to test our framework sensitivity to different genetic architectures, we did additional simulation, varying the number of KTL and adding genotype by environment interaction. Actually, it's genotype by ear interactions. As a result, we saw that the framework will depend on the ability of the fitted statistical model to capture the genetic complexity of analyzed trial. So here we have simulated an additive trait with either 10, 100, and 1000 KTL per chromosome, which give us 210, 2100, and 21,000 of KTLs per genome. In addition, we also add variation in KTL effects across year to account for genotype by interaction. So in total, this gave us just 6 scenarios, where in the first column is the scenarios without interaction, and in the second column, scenarios with genotype by interaction. Here for genetic variance and here for genic variance. For scenarios without this G by Y interaction, the estimates of genetic and genic variance were very much in line with the true values, so we can see that the dots overlap nicely this red dashed line. And it was also largely insensitive to the number of KTLs. So we increased or reduced the number of KTL, and this doesn't change much in this genetic and genic variance estimation. For scenarios with genotype by interaction, we can see substantial overestimation of genetic and genic variance. This overestimation decreased as the number of KTL increased. As summary, we can say that genotype by year and also genotype by environment is clearly an important factor, and we need more research into this. But I also would like to emphasize that we simply need better statistical models to take this effect into account, and then our framework can be applied to it again. Well, as next steps for these studies would be include estimation of non-additive genetic effect and non-additive genetic variance, dominance, and epistasis. Also study trends in locus-specific genic variances, study partition of genetic variance between and within families, and understand better the influence of population structure and admixture between populations on genetic variances. Well, every simulation studies have assumptions. For this study, we are simulating an inbred breeding program, so consequently we assume only additive genetic effects. We could also assume epistasis additive by additive effect, but we wanted to focus in the main effects, so we accounted only for additive effects, the main additive effects. We also assumed a sufficient dense number of markers that collectively capture variation of the trait, and we assumed that allele effects are constant over time and across group of individuals. As conclusion, we demonstrated that this proposed framework works well for genetic partition, but the ability of our framework to capture this varying KTL effect will depend on estimation step, as will any other statistical approach proposed in the literature. We observed a reduction of genetic variance due to selection and drift changing allele frequencies, and due to selection inducing negative linkage disequilibrium. To finalize, I would like to thank this amazing group that I was lucky to be part of, and thank you all.
Thank you for the nice talk, Leticia. This was really interesting. So for anyone who wants to ask a question, so either write it down in the chat box, or you can, oh, this is Zoom, I don't know where you can raise a hand. I think you can. And you might also unmute yourself and just ask the question. And so there are no questions yet. I would just like to start. I think I start from the back because I have multiple questions. So when you talked about model assumptions, you were talking about a sufficient number of marker or a sufficient marker density. Excuse me, sorry. Is there like, so because you tested for 210 or for 400,000 and so on markers, did you also go higher with variance or the markers you tested, and was there like an optimal number for where you didn't found an improvement of the estimation anymore?
This 2,100 actually it's not the number of markers. This is the number of QTLs.
Sorry,
yeah, that's fine. For the number of markers, we had 10,500.
Yeah.
If we didn't test change this number of markers, but the risk of like doesn't have a dense number of markers would be that some effects of the genetic variance would not be captured in the analysis.
Okay, sorry, I hold on with my questions for a second in case there, that was much more. So I start with Thiago. So he asked whether there's any result that particularly surprised you.
Initially, I was not expecting some bias when count for interaction, genotype by year and genotype by environment interaction. But the main surprise for me was seeing that this between linkage disequilibrium, it's a very important factor to change genetic variance, because we assume that the linkage disequilibrium within a chromosome would be higher because it's linkage with the QTLs. But if we see here, maybe you will have a better idea. If we just check one dot in this matrix and compare just one dot, we can see that this within linkage disequilibrium, actually it's higher than the between linkage disequilibrium. And this maybe it's because why a lot of people just won't worry about this linkage disequilibrium per chromosome, but collectively, when we sum up all of these dots, because of so many dots here between chromosome linkage disequilibrium, at the end, this linkage disequilibrium it's much more affect much more the genetic variance than the other the within linkage disequilibrium. And in my passing studies in the past, I had studies with linkage disequilibrium that I don't know, because it was more easy for me that time, I just analyzed it within chromosomes. But for now, I have a different view. So this is also surprised me quite a lot.
Yeah, I agree. So there's a question from Ali Reza. I think you can leave this because I think he asked about this slide. And so he asked, what are the black bars, or what do the black bar means in the graphs shows in the slide? So I think he's talking about this particular slide you're showing right now.
Yes. Okay. So just black lines represent the true value, because in simulation we know the true value and we have the estimated value. So we want to compare if our estimated value, if the distribution of our estimated value would match nicely this true value. So in all this here, for example, the true value is the full line, and here the true value is the black line. So basically, yeah, this is the true value of genetic and genic variance, and this is the distribution of the estimated genetic variance values.
Yeah, thanks for the clarification. And then Anik asks, so she says, thanks, Leticia, for your presentation. How does your estimated across chromosome LD variance compare with other estimates in literature? Is this variance expected to come from epistasis and or potential other things?
No, we didn't account for epistasis here. Yeah, can you read again the question,
Yeah, sure.
Julian, please?
So how does your estimated across chromosome LD variance compare with other estimates in literature? Is this variance expected to come from epistasis and or potential other things?
Okay, so if I understood well, okay, the majority of studies in the literature, they report linkage disequilibrium within chromosome, which is referred to an effect of linkage between loci. Between linkage disequilibrium here that are between chromosomes, it refers to linkage disequilibrium, and it's basically the covariance of these loci for different chromosomes in the genome. I don't know if I answer exactly.
Anouk, do you want to unmute yourself potentially and just ask yourself? And also the second question you have?
Yeah, sure. Leticia, I just wondered if you had an idea where that across chromosome covariance comes from, if that's like epistasis or something else. But maybe like my second question, I find that actually more interesting is like, so you were able to partition the genetic variants in different components, right? Some coming from LD, some coming from other things. And I actually wondered if we could utilize that in, for instance, better managing breeding programs, for instance, and if you have some ideas how we could use that.
Yes, for sure. We can check how these breeding programs dealing with the variability in the population, how the selection would affect the genetic variance. Basically, my just two linkage disequilibrium part. The first one, that is within chromosome, it's due to linkage. The second one, it's due to linkage disequilibrium. Here we are not taking into account epistasis, so I would not say how to link that. We can use these studies to check how the breeding program manage the genetic variance, if they are losing alleles in the breeding programs, how strong have been the selection, and how much variation is there still in the breeding program. So we can do this analysis in several levels of the breeding program. We can do that in time, in stages, in group of families, as said before, and also in genomic analysis as well. Yeah, I think it's that. Not sure. But thank you, Anouk. Yeah, we can also discuss that better later. Yeah, but okay.
Sorry, Gregor just added to the first question that
Julian, are you there?
I think she froze.
Ah.
But yeah, no, so correlation between loci on different chromosomes comes from the selection process that has been induced, that induces this negative correlation, the so-called Bulmer effect.
Am I still frozen?
Yeah, you froze, Julian.
I don't know whether Clemence or Ivan can take over in case I'm still frozen.
No, actually, you're good now.
Okay, sorry. So Gregor just added his comment. And we have another question from Ali Reza, and he asks, in terms of the bias, what would be the expectation if the bias with the size of the QTLs affects and heritability? No. Yeah, sorry.
Can you repeat the question, please?
So he asks, what would be the expectation if the bias with the size of the QTLs affects and heritability of the bias? I think that's what he wants. What would be the expectation of the bias with the size of the QTLs affects and the heritability?
How the bias affect the heritability of the trait?
Sorry, may I ask my question?
Okay.
Yeah, go ahead.
Thank you.
I was wondering when you show the difference in number of QTLs here in the last column in this slide, when number of QTLs are lower, the bias is higher. So would it be a function of because you had only one trait and one heritability? And would it be the function of different traits with different heritabilities also? For instance, if we have a higher heritable trait with low number of big size QTLs or vice versa, how it would be?
Well, I don't know actually. Gregor, can you jump in?
Sure. Yeah, Alireza, I think it really depends, to the statistical model, how well it can capture that effect. Let's say I have just one QTL and, whatever heritability is, if the statistical model can shrink all the markers to, to zero effect and, the closest marker, hopefully it's highly linked to the real QTL. And, if that marker can grab the QTL effect as best it can, then in principle, the method should work well. So, the method that we're proposing here, it's not impacted by, it's impacted fully by, how well the statistical model to estimate the marker effects works or, doesn't work. And that obviously is a very important question. And, we have lots of methods that more or less they all kind of do the same thing, really, if we're honest. So we haven't cracked that nut yet.
Thank you, Gregor. Because in this study we used how do you say, a simplest model, a statistical model, so it didn't account for the bias in the fitted model. So we could not, we didn't explore this variation in the model, and because of that we end up with this bias when we have this genotype barrier interaction. So basically, if we go further and study better models to fit the data, our framework, I believe, would behave nicely, as we can see in the other scenario as well. So this doesn't depend of the framework properly, but actually the statistical model.
Thank you. And you said, that this is going to be added in future research or in future models that you're going to work on this, right? Or Highlander Lab, I don't know where you personally, but
maybe Highlander Lab would maybe want to explore that further.
Okay. So we have one question from Mattea. Mattea says, very nice talk, Leticia. I just assume this analysis was made with plant breeding in mind. So would it work in the same way in animal breeding? Are there any differences between the two?
It worked nicely with animal breeding, and actually we have two papers coming on with cattle So one is from Gerson and the other one from Maria. Both of them worked with Carol, implemented this framework, and also implemented in more traits in multi-trait approach. So yeah, exciting new research is coming for publication.
And can you tell us whether the results are similar or comparable in terms of how good the estimates are, or we just invite them for a different presentation?
Yeah, I would say, if I remember well, it was some time ago that
it's always a bit
worked with data. No, but if I remember well, it matched the analysis behaved very well. The partitioning worked well as well. Maybe Gregor knows remember better than I.
Genes, they kind of work, in both kingdoms the same way.
Yeah.
Yeah.
Perfect. So there are no more questions as far as I can see. So yeah, thank you, Leticia, for this nice talk again and for answering all the questions. Everyone else, thank you for attending. I think the next talk we have scheduled for the 15th of February. So in 3 weeks, we're going to send out reminders and invites again. And yeah, have a nice rest week. Bye.
Thank you. Thank you. Bye.
Have a nice day. Bye bye.